is a biofilm? What's the difference? All right, so we can grow bacteria in the lab. We, nutrient media, a lot of the work on Robert Koch on how you grow it, different agar surfaces, what they need. So we've been growing bacteria in the lab, but we understand now that when bacteria grow in the lab, they are different than if they would grow in nature. In the lab, we have pure cultures. You're learning about that in your labs and stuff when we talk about pure cultures and you have only E. coli, but that's just not the real world. The real world, you've got all different types of microbes living together, competing for nutrients, competing for space. And so we have learned that they actually live differently than they do in the lab. And we see things in nature that we don't necessarily see when we're growing them in pure culture and lab. So one of these things that we see about bacteria growing in their normal environments out in nature is that bacteria are capable of forming what we call a biofilm. Now, a biofilm is defined as a community of cells, different species, so not just one species, but a lot of different species, all living together, attached to surfaces, attached to each other by means of those glycocalyces and those fimbriae that we learned are structures used for attachment. Biofilms are important because they lead to in developing countries, they lead to most of our diseases. Examples of biofilm include those plaque on our teeth. We've talked about that before, that the teeth have a solid surface. Bacteria come in, they attach to them, they start growing, which you can easily brush away in the first few hours, the first 12 hours or so. You can brush them away. Once they have lived and replicated, they start to behave differently. They actually express different proteins and they form different structures and they form this plaque, which is a very tough surface that can't just be brushed away, but actually have to go in and be scraped or sonicked and really work to get a hold of that. So plaque on teeth is an example of biofilm. And again, there's many different microorganisms that make up that community. When you see slime form in river rocks, if you see soap scum in your shower, you know what I'm talking about? If you clean your shower every day, not a problem. You let it go, and then suddenly you get this scale that you can't get off. This soap scum, that is bacteria that has formed a biofilm in the shower. This also happens in implanted medical devices. A catheter, we insert a catheter into a patient, and then that becomes a solid surface that bacteria can implant on and then lead to diseases. All right, so biofilms are very important. So since they cause us problems, we need to understand them. And I mentioned that in industrialized countries, 70% of our bacterial diseases are due to biofilm formation. Dental issues, kidney infections, those types of things. All right, so why do the bacteria do this? Well, there's a lot of advantages to the bacteria for living in a biofilm. Just like for humans, we like to live in communities, and there's advantages to that. That's why I keep pushing those study groups when we make those communities. There are advantages to that. There's advantages to the biofilms as well. So when bacteria are living together in a community, they're able to share nutrients. They're able to protect one another. So you've got an outer layer of bacteria and you spray some bleach or something on it. The inner layer is protected. So protection is a big advantage to bacteria living in a biofilm. And again, it's not about the individual, making sure that individual bacteria survives. It's making sure that the population survives. They can share enzymes with one another. So we talked about exoenzymes when we did our enzyme lectures. Those are enzymes that are secreted out into the environment, which means other bacteria can use them, which means other species of bacteria can use enzymes. One species of bacteria does not produce this enzyme, but it's living next door to a neighbor that does. It's an exoenzyme. So now this bacteria has access to an enzyme that can do something, break down food, do something beneficial that it couldn't make on its own. So sharing these digestive enzymes is an advantage for bacteria living in a biofilm. And then we've already mentioned that they do act differently in groups than alone. All right, so this is some of our a diagram to see that what happens is there's a solid surface and bacteria land on that. You can see different types of bacteria are landing. Oh, sorry, I keep pushing the wrong thing. Different bacteria are landing, and then as they attach, and then once they figure out that they're with a community, they will start producing different proteins and different molecules. So that changes their shape, their biochemistry, and you can see that in the second version here of all these different types. These bacteria look different than when they were living as individuals. And then they can do those benefits that we talked about, sharing water, sharing nutrients, all of that kind of stuff. Now, some molecules can leave the biofilm and they'll go colonize another place, and so that happens too. Okay, so that's biofilm formation. So how does the bacteria know it's in a community? How does it count and figure out the density of microbes and know that it's living in a population, and it should start making these structures that allow it to share? They don't have eyes. They can't take a census. We're all completing the census for the United States right now. They can't do that. How do the bacteria know? And they know by this process called quorum sensing. So that's what we're going to talk about next. Quorum sensing. How do these bacteria know that there's enough bacteria surrounding them that they can start living as a community instead of living as individuals? So quorum sensing is defined as the ability to respond to the density, so how many, of nearby microbes. They do this by sensing secreted chemical signals. As population increases, so do these chemical signals. So each one of these bacteria individually is capable of secreting these chemical signals. So, they make a protein, they secrete it in the environment. So here is a low dense population, just a few microbes, and they're secreting their signals. Now, each one of these bacteria also have receptors, and that's what's present here inside. They also could be external on the outside of the membrane. But each one of these bacteria also have receptors. And as there becomes more signals, there's more chance that this receptor and that signal will come in contact. So when you have a high density population, you have lots of bacteria secreting these red signals, there's a lot more chance that some of those red signals are going to come in contact with some of these signal receptors. Now we have communication. Now this bacteria knows that there must be a lot of other bacteria nearby. When that happens, it triggers a new metabolic pathway, a different path, and they start making different structures, and they can change the proteins that they make and turn into these biofilm type formations. All right, so it's about turning genes on and off, and that is in response to these chemical receptors. So that's what quorum sensing is, determining whether you're in the middle of a group or if you are standing alone by yourself. So that's what's going on here. All right.